Okay. So hello everybody, and thanks for inviting me. I'm glad to present some results of my study in the Sonoran Desert. Now, doesn't work. Why not? Oh, yeah, okay. I can do it this. Okay. Yes, so all of you have been in the Sonoran Desert. I think they will share my impression that it is exciting habitat, exciting ecosystem. And in particular, this spectacular cacti, they are so amazing. It is nice just to see them, and it's much more nicer to study them. And there are several of these cacti species which are considered as keystone species. Some are branched and others are like columnar-like. And the most famous one is for sure the saguara. It can get really, really old. 125 to 170 years old. And it's found all over in the Sonoran Desert and other deserts in Northwest Mexico and Southwest United States. Why are they considered as a keystone species? Well, these cacti provide water, food like pollen, nectar, fruits, and seeds for other animals. They also provide shelter, perching place, nesting habitat. Overall, for the saguara, more than 100 species of birds, mammals, reptiles, and insects have been recorded. But one reason that this cacti can provide this is they need the help. They need the help of an engineer, an ecosystem engineers, and these are the 3 species of woodpecker in that area, which using this cacti as a breeding place, and they're making holes and cavities. And these holes are then used for several other species. Not only that the 3 woodpecker species breed in it, also a lot of other birds, including 2 species of swallows, owls, wrens, flycatchers, use them. But it's also a nice place and hiding place for chipmunks and bats. And sure, there are also arthropods in it and cimicides as well. And I will present my study or our study for searching for the cimicides in this kind of holes or cavities of the woodpecker. And okay, even if it feels like bringing holes to the people of Athens, or like the Englishmen say, bringing coal to Newcastle, I would like to introduce shortly the Cimicidae once more, even if I guess all of you know. It is a monophyletic, well-established family within the Heteroptera suborder. Also, the 6 subfamilies are more or less well-established. We have more than 100 species. Important here for this talk is all of them are ectoparasites, and they feed exclusively on blood of warm-blooded animals, mostly birds, bats, but also on humans. And what is different from other ectoparasites is, apart from the moment they are sucking blood, they do not stay on their host. All species are wingless, and that means for dispersal, they have to travel with a host. So we have 100 species and a little bit of morphological variation, but as you can see here, all of them are secondary wingless, have relatively long legs, and kind of looks adapted to be ectoparasites. Okay, what did we study? Okay, we wanted to know what kind of species can one find in this cactus nest cavities in the Sonoran Desert, which species are around and how dominant they are. We also did a little bit of morphological investigation, but I will skip this for this talk. we did demographic studies of nymphs, sex ratios, and so on. And for the cavity, we try to find out which ecological parameters are important for this bed bugs to occupy this woodpecker holes. And we did also some microclimate studies. Finally, we try to find out which host has been used by the cimicids in the cacti using their blood meal remains in the gut. I hope all these things will be soon published in the Southwestern Naturalist. But moreover, we also did some population and family genetics, which is still under preparation. Yes. So what you can see here is our study area. So northwest of Mexico in the state of Sonora, and these white spots or dots represent the cacti we studied. It's over 20. Some of them are not shown here because we didn't have coordinates, but there were also no cimicids, so no problem with this. Okay, here are some pictures from the study and the working from the field work. And as you can see, it is a destructive method. So we opened this woodpecker hole and completely took out all the things were inside. We were making sure that we did not use any hole which were used as a breeding site in the moment we were there. And if one thinks, okay, now we really damage this cacti, well, it is like one reason why this cacti cannot or will die is actually the overuse by woodpeckers. If they make too many holes in this, the whole reaction of this cacti is making like a wood structure as a response, and that prevents then further on the transport of water within the cactus, which can lead that they die. So we just were part of this natural system. Okay, then we take out this sawdust-like sediment in the bottom of these holes, and we try to count and find and collect all cimicids. And the left corner of both, there are also some eggs found. We didn't count them, just as a record. And if you are good, you see a bug in the center of this sediment, and it is difficult to spot, and they do not move, at least one of the species. So it's a little bit challenging to find them. Okay, yes, so we found two species. One is Hesperocimex sonoriensis, a species which is distributed in the Sonoran Desert and which also is kind of restricted to live in this woodpecker cavities in cacti. The second species is Cimex incrassatus, which has a little bit wider distribution from the southwest in the US, northern Nevada to New West Mexico. From a faunistic point, none of our records were new. They all have been found in that area before, but they haven't been found for 50 years. So it is was interesting to find this species, even if it's faunistically not so interesting. Using this fresh material for one species, we provide the first barcode sequence. Okay. So just a little bit of stop. Okay. From Hesperocimex, we found 164 specimens, and as you can see in the figure down there, the most of the holes did not have any bugs. The other, it varied between 4 and 44. per hole, and the mean was about 14.7. The sex ratio was varying from 1 male to 9 females, up to nearly 1 to 1 in one hole. Overall, we found 40% of males and 60% females. But the most of the specimens we found were nymph, and most of them were in a mid-stage larval development phase. Okay, let's see. The second species, we didn't find so much of Cimex incrassatus, just 20 specimens. We found then in five out of the 20 cavities, 18%. And the number varies from 1 to 14 per occupied cavity. And we found nymphs only in one of the cavities, and also mostly have an early development stage. Okay. When we investigated the gravity, we checked or measured the following parameter. The depth of the cavity from the lower margin of the entrance hole, the compass direction of the entrance, the height above ground of the cavity. And for this, I also have to say, it was also seen here in the picture. Holes very high up we didn't reach. We also did not study. We collected this this material in the cavity, and we tried it and weighed it later on. And we did some temperature measuring inside and outside of a cavity. Okay, to the results. It's going to be out that none of the environmental parameters explain the presence or even the dominance or the number of the cimicids from both species or of the most dominant species Hesperocimus in all cavities. The tightest individual correlation was the depth of the hole, something that might indicate for further studies. And the picture here shows you the compass direction of the different holes. And black rectangles mean they were occupied by the bugs, and not occupied or not inhabited are in white rectangles. And of course, the woodpecker decides which direction the hole should be faced. And our study limited to a very tight or very small area. Then you have to keep in mind that this compass direction of the woodpecker cavities varies probably between species. There's a little bit of debate, but it definitely within species varies depending on geographical or climate geographical distribution and on climate local conditions. So generalizations are difficult to make here. Okay, some other results is like the parameters were correlated, which is each other, which is always like, what do we learn from that? It's not quite sure. It's various correlations. So deeper holes tend to be higher on the cactus, oriented toward to the south, and also deeper cavities tended to have more material. Okay, a little bit about this measurement, what we did. So we used the data logger and measured the temperature inside the cavity and outside of the cactus. And what you easily can see is like during night, the ambient temperature drops below the temperature in cavity. But I think what is more important for the bugs is that during daytimes, the temperature of the cavity, like here, is below the ambient temperature. And that is, I think, is important because temperatures far above about 30 degrees leads to sterility, at least in the common bed bugs, and kind of thermo control as part of the pest control and bedbugs. It's sometimes difficult to apply, but it has been used. So I think that is an advantage for the bugs to have a more moderate, not too hot temperature in the cavity. Also, it has been shown in former study that the cavity has a higher humidity. Even if bedbugs tolerate quite a bit of dryness, even dislike it if it's too wet. I think to have some humidity is definitely an advantage also for egg development and the nymphs in a desert area. Okay, the next part was the question, okay, what are they feeding on? So what we used the gut from ethanol preserved specimens. And if the gut or the abdomen was colored in dark or reddish color, then we assumed that it has had blood meals recently. And it's going to be out that the majority, both of the males with 70% and the females 38%, and 93 of the nymphs had recently got blood meals. And that, I will come back to that, is a little bit surprised because we didn't collect them in nests with chicken. So, okay, the next step, what we wanted to know was, okay, what did they feed on? And we did a Sanger PCR. And here we were a little bit surprised because we used specific cytochrome B primers. Also, we tested others for vertebrates, birds, and mammals, those which has been used in studies of mosquitoes or triatoma, but it turned to be out it was not so easy with most of the primers. And the only cytochrome B primer which worked was those for woodpecker DNA, which was nice. So 12 of 22 Hesperocin species tested were positive for woodpecker DNA. But since we didn't get any results with Cimex incrussatus, and we were wondering if they didn't have used any other vertebrates than birds, we sent the material to a company which did a next generation sequencing using what one called a mini barcode sequence from 100 base pairs of the 12S RNA, which can be used both for birds and mammals. And using this approach, we will be able to detect bats as hosts and have some host analysis also for the Cimex incrussatus species. And this was nice because we found, and we come to that in a second, new host and confirmed assumed hosts. Okay, the host of Hesperocimex. Wait, I have to. Okay. The three woodpecker species, which are the engineers, as I mentioned, all of the three has been confirmed or found as hosts for Hesperocimex. Why is it so difficult to say if it's confirmed or found new? I think we found it as a new species because by now the conclusion was, okay, they have been found in the cavities of these woodpeckers, but it was assumed that they use different bird species as a host. For example, the purple martin. So sometimes this was a kind of an indirect conclusion. Yes, they are on this woodpecker holes, but they probably use different species, bird species breeding there as host. We found out that all of three are the host of Hesperocimex. On one cactus, we also even found blood from two different woodpecker species as hosts, showing something which is known, that the different woodpecker species use the same cavity as a shelter. We could confirm that this pygmy owl is one of the hosts, which also has been found in 2005. And as mentioned before, I think it is interesting to say that since we use the holes with non-breeding birds, the birds using the cavities for shelter is facilitating the survival and the development of the cimicides. That means they are less dependent that in the hole where they are sitting and waiting for the next breeding event, they are maybe not so depending on this breeding events as long as they get regular visitors. Even though as many bedbugs, they can survive long starvation periods, over 200 days, shown by Rügman. And also remarkably for cimids, the females lay fertile eggs even without having a blood meal. Usually, the cimicids start laying egg after blood meal. These two characters show that they are adapted to long periods without having any hosts. What I think what our study shows is that this is independent also from breeding events. Okay, let's see. Okay, the host of Cimex incrussatus we found is a well-known host. It is the big brown bat. It is also known that Cimex incrussatus is in such cavities, but it is not so sure if it was known that also the big brown bat was using this cactus cavities. A nice result is that one individual of Cimex incrussatus had sucked blood from an unidentified woodpecker. So that would be the first record from a non-mammal host of the species. Why is this woodpecker unidentified? That has to do a little bit with this mini barcode sequences, which are too short in some cases to distinguish between different woodpecker species. Okay. Being in between this cacti, we thought, okay, this reminds us of a study which has been done in cities about the colonization of buildings on blocks, colonization by Cimex lectularius. This study, which has been done in London and also I think repeated in 1 or 2 American cities, shows that the general picture for Cimex lectularius and cities are single female colonizations. The individuals from one flat are related, sure. But already the individuals from different floors or different buildings are not related, let alone individuals from different parts of the cities are also not related. So the conclusion which Fontaine and Booth did, he said, okay, we have many independent colonizations, and often this colonization is by a single female, and this population has to go to a genetic bottleneck, and we have a high inbreeding effect. But it also has been shown in other studies that the human bedbug can cope very well with inbreeding. There's no disadvantage with respect to lifetime, survival rate, or reproductive outcome. Okay. So we thought, okay, let's assume this cactus is like a building. The different branches are like a different floor, and the whole is like a single flat. Then we would expect that if we have this single female advanced colonization, as we have in the human bedbug, the genetic relationship within a cactus would not be closer than between cactus. So the different holes in one cactus have different colonization events, and they are all not connected to each other. This would, of course, depend on the host who is transporting the bedbug, like a woodpecker. If the bugs could crawl by its own within this cactus or within this building, these should, then you would expect that they on one cactus with several holes are genetically more related than bugs from different cacti. Unfortunately, I can say the second prediction we could not really test because we didn't found enough cacti with several holes occupied by bugs. Usually it was one cactus had only one hole or one cavity with bugs. Okay, what we did is that we developed 10 microsatellites, and then I did population genetics. I didn't do that, but my colleagues did. And then we had this normal procedure. So we had a micro checker for testing of null alleles. We used the program structure, which does a Bayesian inference analysis for testing different models, like each cactus is one population, or find the most optimal population cluster. And we did test for linkage disequilibrium, and we tested this FES and FST coefficients. Finally, I will come to that later. We also did a family related analysis using the ML-relate program. Okay, down in the spar plot, you could see a result of this Bayesian cluster analysis. So here it has been shown using this Invano method that is assuming that our population is divided by 4 different clusters. If the best results, you could see we have here 6 different cacti, and each color, red, greenish, yellowish, the 2 greens, represent one of the cluster, and it's easy to see they are differently clustered in the different cacti. And this is, in a way, the same result, shown a little bit more, showing the spatial distribution of the cacti. And again, one cacti has a unique unique combination, or unique combination of this different kind of clusters of microsatellites, but there's also exchange between them, indicating already there is some chain flow between this cacti. But the things are not so easy as we hoped, because this fixation index, which is quite low, this FST, which, if it is very high, it will show, okay, every single cacti has its own genetic variance or combination. If it's low, it is an indication for some gene flow. But we also have a high indication for inbreeding. Not a surprise, but that makes it difficult to kind of draw a conclusion from this cluster analysis, because high inbreeding effect violates one of the assumption of population genetics, and then it's the point, how can you draw conclusions from an analysis where the assumptions are violated? So we decided, okay, maybe we will be able, with our data sets, if we cannot use a population genetic analysis, we can do some family genetic structure analysis using ML-relate. So what we hope for, seen on the left-hand side, is a proper, well-structured family tree, which tells us parent-offspring relationship, full siblings and half siblings relationship. We were hoping to get more information about mating, remating of females or males, the same individuals, different individuals. But the reality, the family structures were much more complex. as shown on the upper part. But the most complicated thing or disappointing thing is that we had a lot of uncertainties. I try to give you an example here. My mouse, does it work? Yes. For example, this H76 has a share 50% with the genes with H74. No matter if it's a parent-offspring relationship or a full sibling relationship, which can be tested, let's assume it is a parent-offspring. H75 also share 50% of his genes as a full sibling with H74. But then H75 and H76 should have more than just 25% shared gains. So we found, it's just an example, we found several of these uncertainties in our analysis. Okay. Is the program ML related? Says that the consequences of inbreeding for estimating genealogical relationship or relatedness between the individuals have not been explored yet. So we do not know, again, how can we trust this data with all these uncertainties? Because you have to keep in mind, of course, it just gives you a likelihood for a relationship, nothing more. And is it possible, even if you can simulate this likelihood, is it possible to trust them if one of the assumptions for the program are not explored or uncertain? Okay, the final step we decided to do is then, okay, we just make a scatter plot of our microsatellite alleles using a principal component analysis. No population genetic assumptions, no family heritage models assumed or tested, just maximize the variance. And I just wanted to go a little bit to that. The different color here represent different cacti. And the spots in it are represented the individual found in this cacti. And these circles are the 95% confidence intervals. So what you could see, okay, yes, the individuals in one cacti are very similar with respect to their microsatellites, but there's also a huge area of overlap or with outliers, indicating that there is chain flow between them. Yeah, okay. Okay. What are the conclusions from our genetic analysis? Yes, we have a high inbreeding in the system, and we know that the bedbugs are probably as a group well adapted to this. We have a higher connection between these holes in the cactus of Sonora than between the flats, so to speak, in the Simus lectulario studies in cities. And for me, it's quite clear that has to do that the hosts are more regularly used in comparison to a human system. So to speak, in a building, if Mr. Miller from the 5th floor regularly meets Mrs. Smith from the 3rd floor, and both have some bedbugs in their house, as long as you just meet in a restaurant or go to a cinema, there is no big chance for transmission of these bedbugs. The system is different here with woodpeckers as hosts. They are transporting more of the bedbugs when visiting the different cacti. The role of host-independent mobility within one cactus, shown here with the blue symbols, is still unclear because we could not really study it. Okay. Okay, short outlook. What we try to do next, we try to estimate the gene flow again. We did it, but we wanted to do it again, kind of to estimate the transition dynamics of the bugs by woodpeckers or other birds. We also want to restructure our samples, because as you maybe have seen, this really biased. For some cacti, we have just a handful. For others, we have many, many, many. So we wanted to structure the samples more, getting this bite out, and try a family structure analysis again. So that is our specific plans to come. Not so sure are the next 2 points. Of course, I would repeat this sample in a larger geographical scale. And ideally, I also would like to compare this with a second Hesperocemic species living on the Baja California Peninsula, Hesperocemic conchiminiensis, and compare both study systems. And of course, what I'm going to do is I include Hesperocemics in my further phylogenetic studies. Okay, that's what is left, is just to thank the people that helped me in the lab or with the analyzing of the data or in the field. Without them, these results could not be achieved. Okay. I think that's it. I'm open for taking questions.